Hello students, in the previous part we have discussed about operon. There are two types of operon, and this operon may occur in the prokaryotic cell. Why? Because we know that the whole process in prokaryote will be completed in the cytoplasm. So, in prokaryote, regulation of gene can take place with the help of expressional unit accessory protein that contains these type of gene. And we have seen that there are two types of operon, inducible operon and suppressible operon. In this part we will discuss on inducible operon or Lac Operon. Inducible operon. First of all, or we can say that Lac Operon. Lac Operon occurred in the prokaryotic cell, and it was proposed by two scientists, Jacob and Monod. It was proposed by Jacob and Monod. How is the Lac Operon process can be completed or regulated during this process? How the genes are regulated. Firstly, we have discussed that there are different types of gene with accessory protein. Suppose this is the cell membrane. This is the cell membrane. And just inside the cell membrane, a piece of DNA present. This piece of DNA acts as an accessory protein, and this protein may consist different types of gene. One segment which contains different types of gene, so we can say that it is polysystonic. How these genes are present? This DNA piece or accessory protein, it may consist different types of gene. Promoter gene with inhibitor gene. Again, one more promoter gene, operator gene. These are this one. The rest side is known as a structural gene, and there are three types of structural genes: Z, Y, A. These are the three structural genes. This is the accessory protein which contains different type of gene: operator gene, promoter gene, inhibitor gene, and structural gene. This is the structural gene. These are P, promoter gene, I, inhibitor gene, and again P, promoter and operator. We will identify that P stands for promoter gene. I, not inducible gene, it is inhibitor gene, in which mRNA is produced from DNA and then this mRNA to protein. Inhibitor gene. And O stands for operator gene. And there are three structural genes. And these three structural genes are Z, which synthesize the beta galactose. It synthesizes beta galactose. Y, it synthesizes permeases enzyme. And A stands for trans-acetylase synthetase. Trans-acetylase synthetase enzyme. Structural genes contain three types of enzymes. Z, it synthesizes beta galactose. Y, it synthesizes permeases enzyme, and A, trans-acetylase synthetase enzyme. How is the function may perform? If the outer medium, this is the medium, if the outer medium it may consist no lactose, then what would happen if there is no lactose present outside the plasma membrane or cell membrane? There is no transportation of the lactose into the glucose, because this process transfer the lactose into glucose is a highly energy consuming process. If there is no lactose present, then there is no need to transfer this into the glucose. So, first of all, one RNA polymerase enzyme is attached with the promoter gene. We know that there are the two promoter genes. So, at promoter, all the side of promoter gene we will add RNA polymerase. So, one RNA polymerase attached with this one and other with this one. First of all, RNA polymerase enzyme attached with the promoter gene. What is the role of inhibitor gene? This inhibitor gene changes DNA to mRNA. The process in which DNA changes into mRNA, that is known as a transcription. And then this mRNA changes into protein, and this is called translation. At normal condition, this operon system remains off. So we are discussing about off mode. How the off mode may remain in normal condition? If there is no lactose present outside the plasma membrane, the inside the DNA consists this one like such. One RNA polymerase attached with this one promoter gene, and second one this. As the RNA polymerase enzyme attached with promoter gene, the inhibitor gene changes DNA to protein, the whole process. This may synthesize protein. And this protein, if there is no lactose present, because we have seen that no lactose, this protein in absence of lactose binds with operator gene. It binds with operator gene. As it binds with operator gene, the process of regulation or process of translation stops. What is translation? Process of change lactose into glucose. This will be stopped because this process cannot be taken place. It acts as obstacle. It makes a block the process of the changing glucose from the lactose. At what condition? If there is no lactose present, inside the DNA piece, RNA polymerase attached with the promoter gene. At that time, inhibitor gene changes DNA class to protein. When no lactose present inside the cell membrane, this protein affinity show react with the operator gene and block the process of changing lactose into glucose. No glucose formation can take place. No formation can take place, so it remains off mode. We know that normally it remains off mode. We need to turn it on. How we can change it to on mode? We need to change this on mode if the cell membrane consists lactose or outside the lactose. Another one is on mode. Off mode when there is no lactose present. The formation of protein bind with the operator gene and the process will be blocked. Which process will be blocked? No formation of glucose from lactose because there is no lactose. So it remains in off mode. It turns to be on mode. The second situation, suppose again this is the cell membrane. It contains SS3 protein inside the cell membrane, which contain promoter gene, one with inhibitor, again promoter, operator, and then structural gene, three types of structural gene, Z, Y, A. In second situation, if lactose is present outside the plasma membrane, lactose present. Same here at promoter, one RNA polymerase enzyme is attached here, and one attached with another promoter. This is the condition. If lactose present outside the cell membrane, this lactose may pass out through the cell membrane by the diffusion or osmosis process. Suppose there are some lactose enters into the some parts of lactose into the cell membrane. As this lactose enters into the cell membrane, it divides into two parts of protein. Two parts of lactose. One formed allolactose and second remains same lactose. What is allolactose? We know that inhibitor gene change DNA to mRNA and mRNA to protein. Is that clear? Suppose this is the protein. Same here, protein. But there is no lactose. But in this condition, as lactose passes through the cell membrane, this lactose combined, some part of the lactose combined with protein, and this formed allolactose. What do you mean by allolactose? Lactose combined with the protein. That type of lactose is known as allolactose. Why this protein cannot be reached here? Because we know that protein show higher affinity react with the lactose rather than operator gene. In absence of lactose, this affinity show with the operator gene. But as the cell membrane gain lactose, this protein can't reach the operator gene. This will show the affinity reactive with the lactose, and that is called allolactose. If this remains here, the process remains continued because there is no obstacle can take place at the operator gene. Is the operator gene block formed due to the protein combined with the operator gene. The process blocked and that remains off. But here, this protein can't reach the operator gene. This remains lactose. Lactose plus protein is known as an allolactose. So operator gene remains free. In this condition, Z will synthesize beta galactose, Y synthesize with permeases, and A stands for trans-acetylase. Trans-acetylase enzyme. These all are enzymes, and these enzymes convert the lactose into glucose and galactose, in which energy is produced, and we know that if the process remains on, this is responsible for the growth of a prokaryotic cell. Normally condition it remains off. Why it remains off? Because here protein can't gain lactose. This bind with the operator gene and the process will stop. In this, if the lactose present outside the membrane, this lactose is induced or insert into the cell membrane by the permeases enzyme, because permeases enzyme provide a permeability. As lactose enters into the cell membrane, the protein synthesized show high affinity react with the lactose rather than the operator gene. So protein remains here. No reach the protein from to the operator gene, which from the allolactose, the process will remain continued. In this process, the structural gene which synthesize different types of enzymes. Suppose Z structural gene synthesize beta galactose, Y synthesize permeases enzyme, and A synthesized trans-acetylase enzyme, and these enzymes combined change the lactose into the glucose. Suppose 100 molecule of lactose present here, and all of enters into this one. One change into allolactose and 90 change into remains only lactose. What do you mean by allolactose? Lactose plus protein, that form the allolactose. Firstly, these three enzymes digest this lactose. Digest this lactose, change into glucose and galactose. And at last, these enzymes after this one also digest the lactose of allolactose. So there is no lactose present. No lactose present, and that condition this protein will bind up with the operator gene. Process again begins off. Clear? This is the Lac Operon system, because we know that the lactose which synthesizes in the cell will change into glucose and galactose. This process is highly energy consuming reaction. So in the presence of lactose, this process will be occurred, otherwise it remains off. We have already discussed inducible operon or Lac Operon normally remains off, it turns to be on. In next part we will discuss on suppressible operon.